Where are the blood samples from the early epidemic? Chinese leader of the World Health Organization China Joint Expert Team Liang Wannian answered this question at a press conference on COVID-19 origins tracing. Here are 4 things we learned. Firstly, where are they? The blood samples collected during the early phase of COVID-19 outbreak in China have been kept in Wuhan Blood Center in case of medical dispute or lawsuit. The stored blood samples known as blood braids are a small amount of plasma and serum kept in a pilot tube of a blood bag, which is sealed at both ends. Secondly, why can we not use them for research now? Because in accordance with the Article 31st of the measures for the administration of blood stations, the term of preservation of a blood sample shall be 2 years after the whole blood of the component blood is used. A quite small amount of blood is preserved during the period, and it can only be available to respond to possible medical disputes and lawsuits due to the blood transfusion. Thirdly, will the stored samples be the future research work in the next stage? Yes. As the WHO China Joint Panel and experts from the Wuhan Blood Center discussed in Wuhan, the test of the stored samples will be the future research work in the next stage. Once the blood from the Wuhan Blood Center is used and after the 2-year validity term, meaning the samples meet the requirements stipulated in the measures for administration of blood stations, our relevant work will be carried out. Liang Wannian also said to trace the earliest COVID-19 case, testing of early samples, especially the blood samples, is conducive. China is already preparing for the work, including organizing related experts and institutions. So far, it has made several assessments and evaluations on the testing methods and action plan, which will be implemented after the expiry. Lastly, he emphasized that the earliest reported case is an index case at most and doesn't mean it is the primary case. The primary case refers to the first human who was infected at the moment when the virus jumped from an animal to a human. The region where cases were firstly reported doesn't necessarily mean it is the region where the virus jumped the species barrier from animals to humans.